like today, self-guided study materials that have carefully been designed based on practical assignments for an immersive experience, proven to be effective for thousands of students, researchers, and faculty like you who have already completed our Omics Logic training programs. And thank you, Elizabeth, for sharing about yourself here. Now, I would like to invite Ilya Brodsky, our CEO at Pine Biotech, to take today's session forward. Over to you, Ilya.
Thank you, Deepsa. Share my screen. So as Deepsa mentioned, in today's session, we're going to talk about metagenomics. And what I will be talking to you about is bioinformatics analysis of metagenomic sequencing data. So the methods that we'll talk about today, they include the processing, analysis, and visualization of metagenomics data. And so we'll talk a little bit about some more detailed tutorials that you will get access to after this session. But today I will just showcase some examples and hopefully you'll find them interesting. Some of the approaches that we'll discuss in the overall program are processing pipelines, including KIND2, Data2, and a popular package in R called Phyloseq. But today we'll see several examples of how to do those, but also talk about the application. Why are we doing what we're doing? And what are some of the interesting insights that we can get to understand microbiome and metagenomic data? But before we begin, let's see how sequencing technology has changed the way that we can study microorganisms and how specific metagenomic or microbiome data analysis tools were developed for studying communities of microbiome, of microbial communities or microbiota, to characterize them in terms of their diversity, species richness, and differences between different groups of samples, which allows us to ask specific questions and associate microbial diversity with health and disease. So since the discovery of the microscope, humans have been learning about microorganisms that surround us. And we knew that these microorganisms could potentially be dangerous, causing disease. As the resolution improved, we started to understand the unseen world that is so much richer than what we have previously imagined. The scale of optical resolution today can reveal details of up to 1 micrometer, which is a typical size of a bacteria. So you can actually see in modern microscopes what these bacteria look like. These microscopes today are tremendously powerful, allowing us to see smaller and smaller elements and reveal how the world of microorganisms functions on every imaginable surface and actively interacting with each other. So let's take a look at what does that actually look like. Here we see a super high resolution image achieved with helium ion technology that shows the process of bacteriophages in green attacking the E. coli bacteria, which is in blue. So you can see that these bacteria are covering the surface unevenly. They are of different shapes and sizes, and they constantly interact with these other types of bacteria, but also viruses. So the connection between disease and microorganisms that has been established in the 1800s has been changing over the years. And in the 19th century, laboratory methods for growing microorganisms in a lab culture were developed, helping characterize them with staining. These studies provided important information about bacterial function when studied in isolation. However, it was soon apparent that most microorganisms do not live as isolated species, but in fact live in complex communities. Many members of these communities are just impossible to culture in isolation. So today, we know that the human microbiome, or the community of bacteria that lives within and on different surfaces of our bodies, are not just unicellular organisms living alongside each other, but instead, they form highly regulated, structurally and functionally organized communities that are attached to surfaces as biofilms. and interspecies collaborations as well as antagonisms that contribute to their ecological stability. So because of sequencing technologies, like other omics, like sequencing, genomic, transcriptomic, metagenomic, and other types of omics technologies, now we have enormous databases of data about such communities. And these databases allow us to study the data about these communities using computational methods and understand the 99% of uncultivable organisms in the context of their natural environment. So what kinds of environments can we actually study? These communities are extremely stable, even on exposed surfaces like the skin. So many of us probably know of such conditions like eczema, acne, or dry skin. Forming distinct communities, these microorganisms are interconnected and determined to maintain their community. So they're very resistant to change. Bacteria within a biofilm can communicate with each other by producing, detecting, and responding to small diffusible signal molecules in a process called quorum sensing, which confirms benefit for host colonization, biofilm formation, defense against competitors, and adaptation to changes in the environment. Quorum sensing activities in biofilms are also involved in the virulence and pathogenic potential of bacteria and are therefore an important factor in understanding and controlling bacterial infections, as they enable microorganisms and biofilms to become more tolerant to host defenses and antimicrobial agents. So inside each community, there's this balance of bacteria, fungi, and viruses that control species abundance and activity. So we can study changes to understand why do they cause specific conditions and ask questions about which members of these communities are critical for change. But also other places in the human body contain a lot of diverse but very defined microbiota. The human microbiota is estimated to be around 10 to the 13th to 10 to the 14th of microbial cells, with around 1-to-1 microbial cells to human cells ratio. Previously, it was thought that there's a much greater number of microorganisms, but these figures have been updated as late as 2016. So it's a rapidly developing field that has adapted to newer studies that show what's really going on. So these numbers are derived from the total bacteria cells in the colon, the organ that harbors the densest number of microbes. However, each one of these microorganisms carries important functional properties in genes that contain genomic information or the recipe to make proteins that are functional. These properties and the genomic information about microorganisms have been of growing interest to the scientific community that have been resulting in over 80,000 publications over the years. So the majority of these studies have focused on the intestinal microbiome, but we know that other microbiota have been studied well as well. So since the 1990s, microbiome studies were significantly advanced through such major initiatives or large projects as the metagenomics of the human intestinal tract and the subsequent human microbiome project. These are huge studies that involve many, many thousands of samples that have been made possible through the advances in metagenomic sequencing. As a result, we now know about skin, lung, gut, and even brain microbiota that is starting to be characterized, and we can start studying variation associated with specific conditions and disease. Each new project like this represents vast opportunities and challenges to determine which microorganisms play key roles in the development of certain environmental conditions needed for healthy function of human organs and which can be harmful to it. In the past decade, more than 1.7 billion has been spent on human microbiome research. Major projects are today underway in the United States, the European Union, China, Canada, Ireland, South Korea, and Japan. This investment has confirmed the importance of the microbiome to human health and development. So it is now known what this microbiome function has to do with. Let's take a look at some of these functions. we now know that they contribute to critical metabolic, physiological, and immunological functions, including differentiation and maturation of the host mucosa and its immune system, food digestion and nutrition, energy generation, metabolic regulation and control of fat storage, processing and detoxification of environmental chemicals, maintenance of skin and mucosa barrier function, development and regulation of the immune system and fine-tuning of its reaction pattern, and prevention of invasion and growth of disease-promoting microorganisms or colonization resistance. So many of these questions are being answered using next-generation sequencing, but also other technologies like mass spectroscopy for proteomic and metabolomic data. Indeed, the next phase of microbiome research lies in the ability to integrate multi-omics datasets that combine next-generation sequencing, mass spec, and structural data with phenotypic information about overall changes in microbial communities and the environment that they create. So to understand these challenges and the changing nature of such data, we need to look more closely at metagenomic sequencing data and ask what kind of information can it provide for microbiome studies. So let me pause here for a second and let me ask you how many of you are familiar with metagenomic sequencing data? So how many of you have been dealing with or have read about or have maybe even generated and analyzed metagenomic data? So if you have ample experience with metagenomic data, please put in 1 in the chat box. If you don't have any experience and you don't know about metagenomics at all, please put 0. All right, well, thank you for your answers. I see that the majority of you are not so familiar with metagenomic sequencing. So let's define what is metagenomic sequencing and what kind of information does it provide us with. So metagenomics refers to both a research technique and a research field. Metagenomics, or the field that can be defined as the genomic analysis of microbial DNA from environmental communities. Metagenomics tools enable the population analysis of unculturable or previously unknown microbes. And this is important because only 1 to 2% of bacteria can be cultured in the lab, and the ability to identify microbes without a priori knowledge of what sample contains is opening new doors in disciplines like microbial ecology, virology, microbiology, environmental sciences, and biomedical research. So at the foundational level, we are talking about sequences that define diversity in the whole population in that sample. So here you can see that as opposed to genomics and proteomics and transcriptomics, which focus on the whole of a single organism, organisms in the microbiome interact and combine their features, their functional capabilities as an organ, so to say. And so we can look at the whole community, which is the genetic material that is sampled directly from the environment that allows us to study how this community is divided up into different species without needing to isolate them in the lab and understand what do they look like as an individual when they're in isolation. So there are 2 major approaches to metagenomics. We can look at environmental or culture-free sequencing. And culture-free sequencing allows us to organize all of the genomic data into operational taxonomic units and then look at taxonomic classification, or how the proportions of different taxonomies can describe the population. The second approach, and many times these kinds of approaches are combined, is we can extract a particular type of a bacteria, grow it in a culture in the lab, and then culture-based sequencing allows us to determine how is the particular member of this community different in terms of its genomic classification. So by the way, those of you that are completely new to metagenomics, I recommend for you to take a look at our course. It's a free course called Introduction to Metagenomics. So let me pause here for a second and ask all of you to please go to this link. If you're still not registered on the EDU T-BioInfo portal, you can register right now. It's a very simple, you can register with your social media or your email. And once you register, you will be getting an introduction to different types of metagenomic sequencing, basic overview of the steps to analyze and extract taxonomic classification from sequencing data, and learn a little bit about the applications of metagenomics in different domains, like the gut microbiota, gut health, and the relationship between the health of the gut and the brain, and some of the other applications. So please take a look at this right now. I pasted the link in the chat. And once you have visited this link, please put number 1 in the chat box so I know that you've taken a look and it's loading for you fine. Okay, in a few minutes, we will talk about some of the other resources that you can start leveraging for free to understand more about metagenomics. So please take a look at this link. And once you're there and you've seen this course, Introduction to Metagenomics, just put in number 1 in the chat box. All right, excellent. Thank you, Ramanth and Shraddha. Anyone else? I'll wait until at least a few people have taken a look. All right, thank you, Dr. Manoharan. Okay, again, this course is free. Okay, excellent. So a lot of you have went there. So you can take a look at some of the topics that are covered, and please take a look if you're not familiar at all with metagenomics. It will help you understand a lot better how metagenomic sequencing is going to be useful. Okay, so what is metagenomic sequencing? Metagenomic sequencing is typically referring to the 16S ribosomal RNA amplicon sequencing, which is a recently developed technique that is widely used to study microbiome directly in its environment. So when we talk about the 16S ribosomal RNA, we're actually talking about the small subunit of the ribosome that is specific to bacteria, because in bacteria, it is 16S, in eukaryotes, it is 18S, and this specific gene has to be amplified using PCR amplification. Now that gene contains hypervariable regions. The regions of that gene that are also known as HVRs, or hypervariable regions, provide specific signature sequences that are very useful for bacterial identification by their taxonomic association. So the benefit of amplification is also related to the nature of sequencing, because this particular gene is about 1,500 nucleotides in length. And so we cannot sequence the whole gene using short read sequencing like the Illumina MiSeq, but we can take the hypervariable regions, maybe 1 or 2 of them, combine them together, and then sequence a lot of samples at a fairly affordable cost. So it's a very cost-effective approach to metagenomic sequencing. Now, once we have those sequences, we can start clustering the different types of 16S hypervariable regions together into operational taxonomic units. We can do this using hierarchical clustering, Bayesian clustering, or phylogenetic analysis based on multiple sequence alignment. Once we have those, we can organize them into groups defined by variation within that taxonomic unit. The taxonomic units could then be mapped onto a database of these different sequences to understand which types of bacteria do they belong to, or how can we classify them using a taxonomy. Now, there are several databases For example, the Ribosomal Database Project, or the RDP, the Silva database, which is one of the most updated and carefully curated database. And there's also a Green Genes, more of the environmental data is in Green Genes. And of course, there's also a whole database of these sequences on NCBI. So what does mapping on these databases allow us to do? It allows us to classify the different types of bacteria that we have to create a proportion based on a taxonomic level. So here you can see classification by phyla. And by phyla, if we look, this is a fairly low level of classification, but we can look at how to characterize these microbiota and study differences between them. For example, here you can see colon that has a lot of proteobacteria, as opposed to skin that is predominantly is actinobacteria, right? So we can look at these kinds of differences and understand how the different microbiota or the communities as a whole are different between each other. Now, more than that, we can also ask questions about other members of this microbiome. For example, the virome, which is a part of the microbiome. For this kind of analysis, we will need to have a different approach to sequencing. Instead of 16S ribosomal RNA, we would have to sequence all of the genetic material in the sample and then map, instead of the bacterial database like Silva, we would use a database of viral sequences. And so we can analyze the microbiome for presence of different bacteria. And there's a specific amplification technique called 16S amplicon ribosomal RNA metagenomic sequencing. Or we can amplify the viral sequences and study all of the genetic material and try to understand whether there's also a presence of viruses. So we now know that if we include all of this information, there's a tremendous diversity that is really hard to comprehend, but using computational methods, we can try piece together this complex puzzle and understand how this community functions as a whole, both protecting our organism from invaders and intruders, keeping specific species under control because of the interaction between these different members of the microbiome, but also sometimes causing disease or intolerance and other types of sensitivities. So it's really about different approaches to metagenomic sequencing that allow us to extract different types of information. Now, sometimes we are not interested in just describing the taxonomy and the diversity in a particular sample. Sometimes we are interested in specific species that might cause disease. For example, we can look at specific members of this microbial community, either extracting particular types of bacteria or looking at specific viruses that are associated with drivers of disease. Now, the microbiome oftentimes plays an important role in suppressing such diseases as well. So let's think about some project examples now that you will be able to analyze data from. So these are the projects from the public domain, and you can find these projects on repositories like the NCBI to really understand how metagenomic sequencing could be used to get some practical examples of analysis. So what kinds of data are out there? So please go with me to NCBI. Okay, so we go to NCBI, and again, once you get to this link, please put in number 1 so I know that you're here. So just put in that link, go to that link, and I will just search here for microbiome. And you can do the same. Okay, so if you can go to NCBI and just type in microbiome right here. Okay, please tell me, once you find it on this page, how many bioprojects do you see on the microbiome? How many? What is the number of bioprojects that you see that have the word microbiome in them? All right, thank you, Kabia. And Shraddha, 10,424, right? So there's a lot of data that you can access from the public domain, and you can search through some of these examples just to understand the diversity of microbial sequencing data that you can find in these bioprojects. For example, role of diet in the reconstitution of the human gut microbiome and its metabolome, or microbial sequencing of compost, or circulating exosomes and gut microbiome drive insulin resistance in non-obese mice exposed to intermittent hypoxia, effects of physical activity. So really we can get such a diverse range of projects and ideas just by looking at the different available data sets that you can start thinking about if you're interested in metagenomic data analysis. Let me introduce you to some of the important projects from here that will allow you to get a great overview of how metagenomics can be a powerful tool to understand the microbial diversity in the human microbiota. So the first example that we'll talk about is called the American gut. Now, the American gut today is a component of the Human Microbiome Project. And it's a huge effort that has access to sequencing from thousands of participants that seeks to characterize what would be called healthy microbiome. So it was not designed to target a specific condition or a disease. It was just open project to allow anybody, a citizen scientist, anybody who wants to submit their samples and get sequencing results that will allow you to understand how your microbiome is composed out of which types of bacteria, but also how does it compare to some other people. So as a result, this is a very rich collection of data, all based on 16S ribosomal RNA sequencing that allows us to understand sensitivities, the impact of diet, the result of activity, different types of exercises, whether there's a high level or a low level of exercises, different types of locations, and even ethnic and genetic background. So really a fascinating collection of data sets, and all of this data could be found on humanfoodproject.com/American gut. So if you're interested in just understanding what types of questions could I ask, first think about what data is available. And this is a great repository for those of you that are interested to understand the human microbiome. This is the place to go and to start asking those questions. Now, another fascinating relationship in the microbiome is that it has such a profound effect on the way that we behave, our levels of anxiety, and other types of seemingly improbable relationships between the gut and the brain. So this is commonly referred to as the gut-brain axis. And in this project, they looked at the change in the gut microbiota in mice through antibiotic treatment, followed by gavage of donor stool from other mice, and to determine if significant behavioral changes are induced. It's a study that was conducted based on donor mice that have been fed a particular diet, and then the donors gave their fecal samples to recipients, and those recipients had a diminished diversity in their own microbiome through antibiotic treatment. And as a result, they were trying to separate the impact of the diet itself from the microbiome. So in kind of schematic way, you can think about the experiment like this. You have 3-month-old males. These males are the donors. So they were fed a specific diet. We know that diet establishes a microbiome. And so that microbiome can be thought of a microbiome that has been specifically representative of that diet. And there are two types of diets, high-fat diet and low-fat diet, or regular chow diet. Then you have here recipients. These recipients were prepared to receive the microbiome samples by reducing the load of bacteria in their natural microbiome. So basically they were ready to receive and establish the same microbiota that the donors provided. Then they were studied for behavioral testing. And so we can compare their behavior. And what's important is that we can see the differences in microbiota and associate these differences with levels of anxiety that have been measured through behavioral testing. Now, how does this process work? How do you actually go through the steps of analyzing such data? Let's take a look at an example together. So please go with me to our T-BioInfo platform. This is a platform that allows us to analyze data using a visual interface. And I'll briefly show you an example pipeline where you can analyze data from this project to understand what are the results. What kind of results are you anticipating to get from such analysis? So please let me know once you have reached this site. Again, it's server.t-bio.info. Once you are there, you don't have to have a login right now. If you do, that's great. If you don't, that's fine. But if you are able to get here, please let me know by putting 1 in the chat box. All right, excellent. Great. So Beepse is going to put in the chat box, for those of you that don't have an account, she's going to put in some demo account information for you to be able to log in. So you can see that I'm logged in. I have my name right here. If you want to have access after we're done with this session, you can just go to access and fill out this form, and we will receive that form, evaluate your purpose of getting access. But for now, those of you that don't have access, you don't have an account, here are the details for you to log in. So you would go right here and would say log in, and you would use the email and the password to get access to the demo account. Okay, so I know that you all put number 1 for those of you that reach this site. After you log in as the demo account or as your own account, please put number 2. So I know that you are logged in and you can follow along doing this little demonstration of this demo pipeline. Okay, so put in number 2 once you have logged in. Okay, so you can submit, but also for now, just put in the email and the password. Okay, thank you, Ramant. So the email and the password that Beepsa just shared in the chat, if you don't have an account yet, use these details to log in and get access to this demo pipeline. So I see Ramant has access, or Ramnath, sorry. Has anyone else? Okay, so Ramnath and those of you that have been able to log in, please take a look at the bottom of this section called NGS data. If you scroll down to the bottom, you will see here Amplicon 16S, 18S. So this is the section that we want to go to. And you can actually click on this arrow that will give you access to the demo pipeline, which means you don't actually have to upload the data. We'll just go through the steps. So let's take a look at what this looks like. So here we have what's called the data to pipeline. This pipeline was designed specifically for filtering out some of the common noise from the reads to identify very accurately what are the hypervariable regions grouped together by operational taxonomic units, and then map them onto a database called Silva to assign taxonomy. And then we'll take a look at an example here using Phyloseq, which is an R package for visualization of the data and to do analysis of data such as alpha and beta diversity. So the way that this works is you can read as you go through the pipeline creation process about sequencing, and here are those hypervariable regions. So in this case, they used V4 and V9. So you can see V4 and V9 right here. Those were grouped together. You can see here 16S ribosomal RNA gene PCR amplification of variable 4 region, so V4. And then amplicon pooling and Illumina sequencing. So now you can click on OK, and let's include the data to section. So the bioinformatics workflow here is focused on accurate sequences because they play a major role in our ability to accurately assign taxonomy to the different operational taxonomic units. And the way it is done is by denoising and then merging, these are paired end reads, to cluster into these operational taxonomic units and remove chimeric operational taxonomic units to assign taxons only to the accurate sequences. Now, once we have those sequences, we understand what are these sequences going to, how are they related to each other? So we can use APE, which is a phylogenetic analysis of those sequence relationships. And based on those sequence relationships, we can build a understanding of the diversity, richness of the samples and compare them to each other. Okay, so if you click on OK, click here on End, you can see as a result, you will get some table outputs, for example, OTUs and their abundance between the samples, as well as what are these OTUs assigned to at different levels of taxonomy to be able to compare them to each other. Okay, so for those of you that have completed this pipeline, you should get this kind of a set of outputs to give you an idea of what you might be getting out from such analysis. So if you have gotten the results, please put in number 3. So again, for those of you that have logged in using the demo account and have gotten the results, put in number 3 in the chat box. And I'll pause here a second, and just so that you can see what the pipeline looks like, you can see the pipeline right here. Okay, Shraddha, were you using the details that Beepsa shared? All right, great. Kabia was able to do the demo. So Kabia, you can start looking at some of the results, and please take a look on these Phyloseq plots. So this PDF right here, you can just click on it. Okay, so Jin Long, if you just scroll up, you will see Beepsa pasted the details. Okay, thank you, Beepsa. Here she pasted this again. For those of you that have completed the pipeline, again, you look at this one, phyloseqplots.pdf, and you can open that up just to see what kind of an output are you going to get. All right, so has anyone else except for Kabia completed this pipeline? Just give it a couple more minutes, and here's the pipeline again. Okay, again, for the duration of this workshop, you have the access to this demo account. So please use it to try to understand what kind of information are we getting from this pipeline. Now, importantly, remember that our analysis is around understanding the differences between two different types of diets. Grouping those samples together will allow us to compare them between each other. So you can see in the results, let's think, what are these results? What do they actually mean? So here you have samples, BKF01, 02, and 03, as well as 13, 14, and 15. So these are high-fat diet, and these are chow, regular diet. And you can see the proportion of their operational taxonomic units organized by taxonomy, by their family, and comparison between themselves. So you can see, for example, that in most of them, the biggest proportion is this orange one, and the orange one is Bacteroidetes, so this one. Now, the second one is this green one, right? And so this plot in itself, as we look at this plot, it is a little bit difficult to understand what exactly are the proportions. And so to be able to logically approach such challenges, this gives us a quick snapshot of how to compare them, but it doesn't necessarily provide us with a very clear understanding of what it is that we want to get out of this type of information, right? Before, we looked at a pie chart that was much more easy to interpret. And that's why Phyloseq is a useful package, but you really need to get to know the data itself and to understand the different types of analyses. You can see here different types of comparisons, alpha diversity, beta diversity, different types of phylogenetic plots. So how do we know how to modify these plots and extract the type of information that would be useful to us? Now I'm going to take you to another resource that we will be covering in detail in this program. And that resource, as I mentioned, this is a package in R. So this resource allows us to learn a little bit of R to understand what is in our data. Okay, so again, bear with me. I think at the end of this process, you will really understand what can you get out of this kind of metagenomic sequencing data. So please go with me to this link. Thank you, Bipsa, for pasting that link. Again, the registration here is free as well, and we will extensively go through some exercises in R during the program that is going to start in November very soon. So please go here and you can register. So again, I'm going to, you can register by get started for free. And once you're in, you can go to a collection of courses that are in both Python and R. And I'll show you a little bit of how to work with the outputs from the data to pipeline so that you understand how to build plots based on taxonomic classification. Okay, so again, for those of you that reached codeomicslogic.com and have signed in, please let me know once you're in and you can see the courses. All right, excellent. Anyone else? Just give here a couple. Okay, Robert, great. All right. So what we want to do here is to understand how to take the outputs from the pipeline and actually use R to try to ask questions about, well, what's in our samples? How do we compare them? All right, so I see a few of you have signed in. Let's go to this course called Metagenomics. So you go into the Metagenomics and you can see here a number of free courses. So we'll go to the second one, visualization of 16S metagenomic data. So by now, all of you know what is 16S metagenomic data. So let's go in here and you will see what this is all about. So here we have the data sets from this pipeline that we just did. So this pipeline includes analysis of the OTUs and their taxonomic level, and here we have it by phylum. So in Excel, what we can do is we can just take the different phyla that are mentioned in here and the abundance of this phyla to then summarize the information and create a pie chart. And that will give us kind of that same figure that we already saw before. So once you're in here, just click on next. Here you have a description of how to do this in R. Okay, and basically it's just a simple function to create a pie chart. And then you can see that we can also measure the abundance using a bar plot. And we can do this for one individual sample. Okay, so here you just go through the code, create a nice looking chart using different colors, click on next. And now let's try to do this together. So once I'm in number 3, try it yourself. We're going to try and do this together right here in this little terminal using R. So let's go through this code really quickly. Here we have the library, open XLX. Here you have sorting the table. So we load the table, sort the table, and then we create the percentages and make a pie chart, right? So a very simple script. And also we can create a bar plot. But here you can see that the instructions say to create using this data set. Okay, so link here, and we just need to replace this little word right here. So just delete it and paste it right here. Okay, and now we can click this button, run. So it will run, and you can see that in the console, it gives us the answer, and the answer is Proteobacteria 49%, Bacteroidetes 24%, Firmicutes 13%, Cyanobacteria 6%, Actinobacteria 4%, and Fusobacteria 2%, and so forth. So now we can see also some of the plots, and we can look at them either by percentages organized in a pie chart or in a bar plot organized by what is the top. So looking at this chart, what is the dominant taxonomy in this sample by phyla? So what is the dominant phylum in this sample? What can you say about this graphic? And what is that? Do you see what is the name of that bacteria? Okay, it says Proteobacterium. Okay, and you can actually see right here that number 1 is Proteobacteria, and it constitutes 49% of the sample. Okay, so Proteobacteria, let's just take a look at what is this Proteobacteria. What does it do? Okay, so Proteobacteria is a major phylum of gram-negative bacteria, and they include actually a lot of different types of bacteria, and a lot of these, let's see. Okay, so you can see a little bit that they're responsible for nitrogen fixation. Okay, and since this is in the gut, we can say Proteobacteria in the gut. And let's take a look at what is this. Okay, the bacteria phylum is often misrepresented in these individuals, with E. coli being the most prevalent species. It is clear that a complex interplay between the host bacteria and bacterial genes is implicated in the development of these intestinal diseases. So it actually is indicative of a common factor in human diseases. And you can see that it is a microbial signature of dysbiosis in the gut microbiota. So it does have an important role in kind of shifting the balance that should be there in the gut microbiota. Okay, so does anyone have any questions about this? Okay, if you have any questions, please put them in the chat. Now, if you look at the overall course, this course includes some more additional types of lessons that we will go through in the course itself by looking at taxonomic composition. This is a more advanced course. You will see that we will look at comparison between exercise frequency, and we will look at much more complex code that you have to learn as you are trying to understand what are the differences between different samples. And you can see here that we will learn how to create all of these different charts and make sure that those charts are representative of the total percentages, and also compare them between groups. So all of these are important downstream steps to be able to ask the question, what is the difference if we identify groups of samples, and how do we know for sure that these are differences that are statistically significant between groups of samples? Okay, so we had here a brief overview of the data-to pipeline, and we understood how sequencing data is processed to create an output. And then we looked at some examples of code in R that allow us to study in detail some of the variation to understand what might it mean and start thinking about the biological implication. Now, what other types of use cases can we find in these publicly available repositories to be able to ask other interesting questions? Well, one important example is shown here, where we can see that the gut microbiota actually has a significant impact on different types of conditions and different types of treatments, the efficacy of treatment. And so this idea of modifying or modulating the microbiota has a profound role in how we can think about improving efficacy of treatment, diminishing the risk of disease, of developing disease over time, improve the overall sensitivity to different types of compounds, and protect the individual, preventing diseases from developing. In fact, there has been even proposed treatments for different types of complex conditions. Like, for example, you can see here in 2019, there was an article that China approved an Alzheimer's treatment that targets the microbiome. So very interesting development in this field. We also know that, of course, many different types of drugs are metabolized through the intestines. And so the intestinal composition of the gut microbiome plays a huge role in how we can metabolize accurately and also develop microbial signatures, which are, of course, easy to sample from fecal and gut samples. And so by understanding the microbiome, there's an opportunity to improve treatment, to improve outcomes, to develop stronger immunity, and really change the way that we work with treating different conditions using different types of treatments. So to summarize, in this program, we are going to be speaking about different aspects of metagenomic data analysis starting on November 17th. This online program is designed to be equally rewarding for students and researchers, and it's especially suited for project-based learning on a flexible schedule. So we'll cover main aspects of different types of metagenomic sequencing, different approaches to analysis of metagenomic data, including whole metagenomic data, as well as 16S ribosomal RNA amplicon data, and other aspects of microbiology research using bioinformatics. At the end of the program, participants will be conducting independent or group projects, just kind of what we looked at today, analyzing data from the public domain and learning from high-impact biomedical publications on the topic of the microbiome. And in a few minutes, Bipsa will tell you a little bit more about the program and how it is organized. Now, in this program, as in all of our omics logic programs, we will highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, machine learning, Our transforming personalization of treatment selection and innovative algorithms allow for unprecedented capabilities with drug discovery, design, and repurposing. The various types of omics data describe in great detail intracellular and intercellular processes associated with common diseases. These data sets can be analyzed to understand the biology behind host factors and microbiome composition in our bodies and the environments that we live and interact with to drive infection and cause deterioration of health, to destabilize the immune system and lead to clinical manifestation of disease. Now, importantly, the objective of this program is to allow you to think critically about different data sets and methods of analysis, present your ability to apply bioinformatics tools to such data, and get feedback both from the community, the participants of the program, but also experts that have experience working with such data to guide you on how to improve your analysis and the presentation. Now, all of our Omics Logic programs are designed to provide students of all backgrounds with a clear perspective on big data analytics, machine learning, and artificial intelligence in application to omics data and the research, clinical, and industry applications of such methods of analysis. And so this step-by-step process allows you to try out many different types of analysis very quickly, understand the inputs and outputs, as well as what critical settings in the pipeline have a huge impact on the outcome. And to test such analyses, not on just data sets that we are going to provide you with, but also to guide you on how to develop an independent project by finding a data set that is interesting to you, that you would like to analyze, and you would like to interpret for yourself. So we actually will have whole sessions on how to do that. Previously, participants of our previous programs have led on to develop poster presentations and publications based on what they have been able to achieve in the course of this program. So the program, again, will start on November 17th. You can find all of the information on metagenomics.omicslogic.com. And to guide you a little bit through the different sessions that we will have, I will pass it back to Beepsa to speak about the schedule and the registration.
Thank you, Ilya, for that wonderful overview. I will start by sharing my screen here in a minute. And I hope that everybody enjoyed the hands-on session so far. So what we essentially do with our Omics Logic programs, and I just wanted to give you all an overview, that the goal of our Omics Logic programs is to provide training in bioinformatics, enabling independent research, guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. So together with our collaborators, we have been building this growing online community of bioinformaticians in training, ranging from high school students to faculty and researchers in top universities around the world. Now, you are already familiar with these two platforms that we demonstrated today, and also a few of the other platforms that we demonstrated. Now, moving on, let me give you an overview of how to register for the upcoming program. So let us go together to the website. So Ilya has shared this link in the chat. Thank you, Ilya, for sharing the link. It's called metagenomics.omicslogic.com. And once you are here on the site, you will be able to see a program overview and also the different sessions of the program. So as you see here, we are starting the program next Tuesday on November 17th. And then the other second session would be on November 19th. And we will finish it off on December 15th. So you can go through these different sessions right here, and you will also be able to see the different dates if you just scroll around here. And each of these sessions would be at 10 a.m. CST. Okay, this stands for correction. It would be at 9 a.m. CST, which is 8:30 p.m. Indian Standard Time. So to register for the program, you can click on Get Started, and you have 3 options. The 3 options are for the 3 different levels that you can apply for. So the beginner covers your access to all of the resources that Ilya discussed today, and I will again be going over very shortly. The intermediate program gives you access to these resources plus a one-on-one mentor support, where you will be able to go beyond these scheduled sessions and get on a call with a mentor who will be able to guide you with your projects or your questions that you have. While the advanced membership option gives you the chance to work on your project for not just this 1 month period, but extend it to another 30 days. So it's like a 2-month access to the program and to the platform and resources, and be able to collaborate and work with also the research interns who are part of this bioinformatics community and part of the Pine Biotech Research Fellowship Program, and work on your own project. So these are the differences between the 3 programs. But if you are a beginner, you can just start with the beginner membership, because you will not miss out anything and you will learn a lot from the program. And once you're registered, the first site that you will have link or access to is the organization page. So from this organization page, you will be able to take courses, projects, and also explore the syllabus that is there. We hope, Sabila, that we are able to do things for free, but these programs, these assets and resources also require a lot of investment on our part. And at this point of time, we hope we are not able to offer it for free for everyone, but there are a lot of free resources, as Ilya also showed. And I'm also coming to the next. So you could start by taking the free courses and utilizing the free resources. And next here, you will have access to all of the premium courses on our platform. So it includes both free and premium courses. So you will have access to them, as well as projects. So some of the projects that already Ilya is describing here will be covered, like the American Gut Project. But then also there are other projects which we will be covering, and these also you can do on your own. Next, you will have access to the T-BioInfo platform. So today, the access that we shared with you on the chat was a demo access, which only allowed you to use the demo pipelines, like we used this one today. But with the educational access, you will be able to utilize the full platform. You'll be able to utilize data sets from public domain repositories like NCBI. And this platform is also available for research use, which many researchers, labs, and universities utilize. So that is also available, but this is all included in the pricing. You're not paying an additional fee for this. And you will also get access to all the free courses that are available in the Omics Logic Code Playground, as well as the premium courses. So there are some courses on the Omics Logic Code Playground which are paid. So you will also get access to those free courses as well as part of this full 1-month training program. And if you have any questions, please let me know. And before I pass it on to Dr. Mohit to take this session and end today's session, I wanted to ask you all to do a little bit of poll for me. So I hope, can you all see this poll on my screen?
Yeah, we can see it on our screens. Thank you, Bitsa.
Yes, and if you would please also put in your answers to this poll, that would be great. Thank you so much. So, Dr. Mazumdar, if you can take over, and the poll can continue running. Thank you.
Yeah, 1 second. Sorry, Mohit, we'll make you co-host. I don't see. Okay, there you are. Yep.
Thank you. Thank you, Ilya, and thank you, Bipsha. So let me share my screen. Hello, everyone. My name is Mohit Mazumdar, and I'm one of the global representatives of Pine Biotech. So in my current role, I'm working with scientists and researchers and students to assist with their omics big data research and collaborating with universities to provide support for bioinformatics training and education. This Omics Logic Metagenomics program is a specialized program that not only takes you through the live sessions, courses, example projects, but also get you to learn by applying this knowledge to a real metagenomic project. So during the last program, we worked with our participants throughout the program, resulting in more than numerous independent bioinformatics research proposals and projects. So these research proposals were then peer reviewed by experts. Some of the research proposals included analysis of microbial ecology of the
Sorry, we
international
don't
space.
see the screen. We just see a black screen.
Okay. So let me share again. Can you see it now?
Yeah, we can see it now.
Yeah, we can see it now.
So I'll not play this. It's having some issues. So as I was telling you that during the last program, we worked with these many participants. Some of them you can see right here. So the project proposals that were proposed after the end of the live sessions. So there's a period where we work with the participants to work on their projects. So the project proposals that we got from these participants, I wanted to give you some examples of those project proposals. So one of the project proposal was about microbial ecology of the International Space Station and implications of long-term human missions to the moon and Mars. And there was another interesting proposal about plant and animal cytogenesis relationship with SARS-CoV-2 virus, toxicity and transmissibility, and how metagenomics can help in this research. And then another proposal was about cutaneous microbiome in patients with filariasis, so about the skin disease. So these are some of the projects and that I wanted to tell you about today. And moving on about the program and about the resources, I think one of the resources is that we would also like to recommend you to visit our blog, where we publish informative articles related to bioinformatics resources, omics logic training programs, and many more. So this is the link to visit the blog. So this is another resource that we would recommend you to go to. Another important resource that I thought you would be interesting to know, and if you're interested to learn about latest research articles, news, job opportunities posted by pharma companies and research labs, and also about our upcoming programs, then please join these social media groups to get regular updates. Yeah. So if Bipsha is sharing those links. Okay, thank you, Bipsha. Great. So with this information, I would like to thank all of you, including our program speaker, Ilya, and Bipsha, for helping everyone. And we hope that you will go through some of the resources and links shared today, including the recording of this session, which we will be sharing soon. And thank you again for everyone, and have a nice week. Thank you.
Mohit, there's a question from Kabir.
All right.
I will read out the question for you, Dr. Mohit. Did you come across any project or group working on microbiome in relation to immunotherapy?
Working on microbiome in relation to immunotherapy. So, Idia, would you like to answer that? Like any project that we have?
Yeah, so we don't have a curated project that you can follow, but an easy way to check for availability of such data is to just search. You remember I shared with you NCBI, so I can just share my screen here. So here, if you just search for bioprojects on microbiome and immunotherapy, you can see a number of projects, a lot of projects. Microbiome of patients under immunotherapy in China, microbiome-derived enzyme modulates response to checkpoint inhibitor and immunotherapy. So there are a lot of projects. So if you're interested in working on such projects, as Mohit proposed, in the context of the program, that's actually, oh, okay. Sorry, do you see my screen? Maybe it wasn't showing before. So you can see if you just search here for microbiome immunotherapy, you will find a lot of different interesting data sets. There's obviously a lot of publications. You can do some literature review first, but after you've identified the specific interest that you have in microbiome and immunotherapy, here are some data sets. And so once you get access to these data sets, you understand how to analyze this data, you'll be able to ask very specific questions and get a sense of what's possible with metagenomic sequencing. Does that answer your question?
Ilya, there is a question from Kusal. If there is any correlation between COVID-19 and human microbiome?
So as Mohit mentioned, actually, one of the participants in our previous program was interested in that. And so here again, COVID-19, the interesting factor potentially would be how does microbiome correspond with severity of disease, with risk to develop disease, right? Susceptibility. So you can see here a number of projects, for example, gut microbiome alterations and longitudinal kinetics in COVID-19 patients, right? So again, you can look at some of these data sets to just get a sense of whether there's a data set that you can ask these questions of, and then read a little bit about how was the data prepared, what is the data showing, to understand whether what you're asking is a reasonable direction, right? And so once you've identified that, depending on your background and your different resources that you might have access to, actually there's there are a lot of patients, so you could probably generate some of these data sets if you had a real research project. So I think that's a very viable and interesting question that you have there. Okay, any other questions? All right, great. Well, thank you everyone for joining today, and looking forward to seeing you in the full program and working together with you to develop these research questions and understand how to apply these tools. So please continue exploring the resources that we shared with you today. We had a lot of participants that learned how to do this independently, and we hope the same from all of you. And so we are really excited about being able to work with you, and Mohit, as well as Harpreet, who is one of our mentors, will be working with you to help you find the right data sets, to design your analysis questions and translate them into specific analyses that you can do. So this is an informational webinar that is intended to introduce you to some of these topics. We provide a certificate based on merit. So if you participate in all of the sessions in our upcoming program and you complete some of the assignments that are given there, you will get a certificate of participation. Or if you excel and you develop a project of your own, you will actually get a certificate of excellence. And so these certificates come from both Pine Biotech and the Tauber Bioinformatics Research Center, which is a research center at the University of Haifa in Israel. So we welcome you to join and get that certificate. It will actually be a valuable certification of something that you've done. Just for participation, though, you can definitely mention their participation in this webinar and share with your other contacts. Hopefully you found this presentation interesting. So again, thank you for joining today.